After the finding of genetic material as DNA, and that is the genetic material, that is the molecule which is transformed from virus to bacteria from the Hershey and chase experiment. But in real means in our living organism, in the living system, it is also found that RNA is also a genetic material, and it can be found in some of the virus, like which is known as tobacco mosaic virus, also QB bacteriophage, and most means in the recent times जो virus बहुत ज़्यादा popular था, like which is COVID-19. Okay, so these all are the iron means or not we can say easily the living organism, but in the living system, these are the means having RNA as genetic material. But how can we say that any molecule can be regarded as genetic material? And for that, we will discuss about some criteria for any molecule acting as genetic material. So this whole topic will be having very less discussion. But okay, so the criteria for any molecule acting as genetic material, the first criteria is they should able to generate its replica or there should be replication. And with this DNA and RNA, they both can replicate their own copy. Then the next one is it should be chemically and structurally stable. Okay. So with this we can know that these are the two molecules which we discussed in earlier discussion also about DNA and RNA. But in this later discussion we will say how DNA we see how DNA is more stable than RNA. Second thing, it should provide slow changes of mutation. Okay, yes, both DNA and RNA can provide mutation so that it will be required for evolution. But how we will say in this regard how DNA is more better, DNA is better for this because there is not that much mutation occurs in the case of the DNA. And in the last criteria, which is DNA and RNA or the genetic material should able to express itself in the form of Mendelian character. And we know about the Mendelian character that one trait should be expressed in next generation into the next generation, or it should be inherited. So with this, these are the different criteria which is giving rise to which material should be genetic, which molecule should be genetic material. And with this, we can conclude that protein doesn't fulfill these all criteria. The criteria rather DNA, but DNA and RNA. And how this DNA is more means in the evolutionary term why DNA is the main genetic material in the most of the organisms that we will discuss one by one with the differences between these two different genetic material. So yes, by looking to the different genetic materials, we can say that RNA is a single strand molecule. It is, it is the case, and we can say here it is this nucleotide contains the pentose sugar is here, which is we can say having 2 prime OH group. And as we know, this 2 prime OH group is present in the RNA. This gives it is due to this it is very reactive in nature. RNA is reactive, more reactive. I'll say here this RNA. Okay, RNA is more reactive because of this presence of 2 prime OH group. Similarly, if we'll say about this DNA, there is absence of 2 prime OH, and that's hence this is chemically less reactive, and more stable. One more example will be this is the first thing. Second thing, we know in the case of this uh, RNA, there is presence of uracil as the base, and in the case of this DNA, there is thymine, and we know thymine has one extra methyl group, and uracil is means absence of this methyl group. So this uracil and uracil and thymine group also confers means having different provide different stability, and this thymine also provide more stability. Okay. Third thing, single strand, and this one is the second one, which is the DNA will be having double strand. And what is the benefit of this double strand or single strand? Because due to this double strand, and we know there is the two are complementary to each other, so they provide complementary strand. So due to this complementarity in nature, if one strand has one particular base and the other strand doesn't have that base, so it will lead to a kind of means इसमें easily कोई एक repairing mechanism हो सकता है ना, so which provides repairing mechanism, and with this we can say DNA does have good genetic repairing mechanism. No repairing mechanism. ओके, तो इसी जेनेटिक रिपेयरिंग मैकेनिज्म की वजह से डीएनए को और अपना मतलब जो स्टेबिलिटी मिल सकता है. देन नेक्स्ट वी कैन से आरएनए less stable and which can give rise to uh, faster rate of mutation. फास्टर रेट ऑफ म्यूटेशन. अब ये अगर फास्टर रेट ऑफ म्यूटेशन होगा तो इससे क्या होगा, इससे एक चीज होता है कि दे कंटेन द viruses which have RNA genome has faster rate of mutation, they have shorter lifespan and they evolve faster. अब इसके रिलेटेड मैं अभी रिसेंट जो कोविड का मैं एग्जांपल देना चाहूंगा. कोविड के एग्जांपल में आपको ये पता होगा कि in the case of COVID-19, there are so many different variants. They arise within some time. हम वैक्सीन ले रहे हैं पर वहां से और कुछ अलग वेरिएंट निकले. वेरिएंट का मतलब क्या? वेरिएशन. वेरिएशन कैसा है? टू दिस म्यूटेशन. बट इन द केस ऑफ डीएनए being more stable, less mutation, and this mutation is means better for evolution. Yeah. And the main thing, which is very much important to the stability, that is less stable than less better uh, form of storage. We know RNA being more means which we can say is more active or reactive, but and it can also use as an enzyme as an catalyst, but still for the storage of information, okay, there is this more stable compound, a more stable molecule is preferred better for storage. More stable preferred preferred for storage of genetic information. Genetic info. But there are some points which we should know, which is very much means, like is a plus point or pro or advantages for RNA. RNA can directly can be transcribed to express create by producing protein. But for the case of this DNA has to rely to be sorry. This would be translated. Okay, can be translated. DNA has to rely to transcribe into mRNA or translation of or expression of protein for expression. Okay, so this will be related to all the differences between RNA and DNA, and why this DNA is being more preferred for stability, and that's why we can conclude DNA is preferred genetic material. ये कोई आके मतलब प्रेफरेंस नहीं दिया है. It is a gradual evolutionary process in the evolution. अब इसमें एक आपके सिलेबस में एक एक्स्ट्रा जो एक छोटा सा टॉपिक दिया हुआ है अबाउट द आरएनए वर्ल्ड. यस, इट इज ट्रू दैट आरएनए वाज द फर्स्ट जेनेटिक मटेरियल. यस, क्योंकि इसी से ये जो बना, फिर देखा गया कि अगर इसमें जो 2 प्राइम ओएच है वो अगर डीऑक्सी हो जाएगा, है ना, वो ग्रुप नहीं रहेगा, तो वो कहीं ना कहीं मीन्स स्टेबिलिटी और दे रहा है. इनएक्टिव मीन्स जो ज्यादा रिएक्टिव नहीं हो रहा, तो स्टेबिलिटी ज्यादा दे रहा है. सिमिलरली यूरेसिल ग्रुप में अगर एक मिथाइल लग गया, तो वो कहीं ना कहीं बन जा रहा थायमिन. थायमिन बनने से मोर स्टेबिलिटी. आरएनए सिंगल स्ट्रैंड था, डबल डीएनए डबल स्ट्रैंडेड बना, कम्प्लीमेंट्रिटी रहा. कम्प्लीमेंट्रिटी रहने से जो बेस पेयरिंग है उसमें अगर कोई भी मिसमैच होने से, जो डीएनए मिसमैच और डीएनए रिपेयरिंग होने से मतलब जो एरर जो भी होगा, तो वो आराम से रिपेयर हो सकता है. मल्टीपल लेवल पे रिपेयरिंग होता है, जो कि आगे की क्लासेस में ये बताया जाएगा. ओके, सो दिस इज द regarding the first genetic material and the most essential जो प्रोसेसेस हैं, है ना, लाइक मेटाबॉलिज्म, ट्रांसलेशन, स्प्लाइसिंग, ये हर एक के बारे में आगे के डिस्कशन में बताया जाएगा. रिवॉल्विंग अराउंड आरएनए, ये रहेगा, है ना, बिकॉज दिस इज द इम्पोर्टेंस ऑफ दिस आरएनए. ओके, एंड ऑल्सो वी नो दैट along with जेनेटिक मटेरियल, इसमें और कौन कौन से अलग अलग फंक्शन है जो कि मैं आपको पहले से भी बताया था, विच इज एज एन कैटलिस्ट, जो कि आरएनए भी सॉरी जो प्रोटीन भी जो प्रोटीन के एंजाइम भी जो रिएक्शन नहीं कराते वो आरएनए करता है ना. कैटलिस्ट और यू कैन से एंजाइम, आरएनए एंजाइम. ओके, ये तो हो गया. उसके बाद जो है देन वी नो, एंड उसमें मैं आपको बता था कि ऑल्सो अलॉन्ग विद दिस जो normal genetic material, they also act as an adapter, right? They can act as an adapter. Also, we can say they can act as enzyme, which I already told about. Okay, so ये हो गया. And adapter and we can say messenger and also an enzyme. तो ये extra functions रह रहे, but RNA being more reactive and less stable, क्या हुआ, एवोल्यूशन के तहत there is formation of, okay, evolution into more stable DNA. एक रिवीजन जैसे भी हो जाएगा. इसमें 3 चीजें हैं. एक डी नॉन एबिलिटी ऑफ 2 प्राइम OH गया, यूरेसिल गया, एंड सेकेंड सिंगल स्ट्रैंड. एंड मैं आगे की कभी डिस्कशन में भी ये बताऊंगा। देयर आल्सो प्रेजेंस ऑफ सिंगल स्ट्रैंड डीएनए। ठीक है, डीएनए के भी सिंगल स्ट्रैंड है। ओके, बट दिस जो सिंगल स्ट्रैंड डबल स्ट्रैंड डीएनए इज मोर मीन्स ज्यादा एबंडेंट होते हैं। तो ये हो गया डीऑक्सी ग्रुप, ये हो गया थायमिन, ये हो गया डीएस और डबल स्टैंडर्ड डबल स्टैंडर्ड and with this they provide more stability. And also there is one more thing which we can say, this double standard, due to this double standard nature of this DNA, there is a process, there is a mechanism of रिपेयरिंग। ये जो जेनेटिक रिपेयरिंग, डीएनए रिपेयरिंग जो है, ये बहुत ही इंपॉर्टेंट टॉपिक है। इतना डिटेल में नहीं है, ये हायर क्लासेस में और अच्छे से बताया जाएगा। सो यही है रिलेटेड टू अबाउट अ वेरी स्मॉल टॉपिक ऑफ what should be the criteria is there for any molecule to be acted as a genetic material, and after that why you know, as we know, these all criteria are being fulfilled by both DNA and RNA, but still why DNA is regarded as the genetic material and why it is present in most of the organism, and RNA is only found in some of the viruses. Okay, and initially we know RNA was found to be the genetic material, but what was there so that this we can say in the meantime, in the evolutionary time, DNA become the genetic material. Okay, so these are all discussion. In the next discussion, there will be very important three different processes will be there. One is the replication, then there is transcription, again transcription, and the last one will be gene expression.